Very good morning, dear students. We are happy to enter into the animal biotechnology. In this animal biotechnology, today we are going to deal with the cryopreservation. We know the preservation, what is meant by preservation, why we have to preserve the material. Preserve nothing but it's a protection. But the term cryopreservation, we call as the cryopreservation. Clearly mention what is meant by cryo. Cryo means it's a Greek word. It means chaos, frost. I mean freezing. In I think heavy temperature. Preservation under the heavy temperature. That is the minus temperature. I think already. Yes, we have seen here. We'll see about the chaos. Is means frost. It means preservation in a frozen state. We know the frozen state. It stops the degradation of the material, which is we keeping the freezers automatically. It stops the enzymatical reactions. This is the process of cooling and storing cells. Cryo, I think the minimum in is minus degrees. When we are using the cryo means the lifespan of material is there. We can use the material which is undergoes the cryopreservation. We can use long lasting, nearly 50 to 100 years after. Also, we can use the material for the further copies and use whatever it may be. Yes, we will go in this cryopreservation several gametes of the yes, endangerous species. We can isolate the gametes of the species of animals and plants also, the roots of plants, which is the rare plant like we collect and preserve in frozen condition. When we preserve the frozen condition, the material and their uh, texture and their capability does not lose. In this cryopreservation, uh, we are preserving under the minus 196 degrees. It's not easy. Minus 196 degrees. That too, we have placed in the liquid nitrogen. In minus 196 degrees, we have placed it into the liquid nitrogen. Yes. In this cryopreservation, several steps are there. We will see one by one. The first is selection of material. It's very important to select the material which is we are preserving. Selection of material for cryopreservation, the selection of proper plant material or animal material is important. Two important factors depend on it, such as nature and density. Any tissue can be selected for this purpose. For example, embryo, meristem, ovules, seeds, etc. These are all important elements to preserve. Yes, we can see here the second one is addition of cryoprotectants. First step is the selection is very important. Second one is add to the cryoprotectants. The chemical material is important as it preserve cryo destructions. Some examples of cryoprotectants are alcohol, some amino acids like prolines and dimethyl sulfamide. Mainly two cryoprotectants should be used together instead of a single one as they are considered to be more effective. What are the use of cryoprotectants? Cryoprotectants are this prevents of a highly frozen. If we not mix the, if we not add the cryoprotectants, the material which is we take for the preservation, the scope is there to attach one to together to form cell lysis, cell death in case. So definitely we have to add the cryoprotectants for the prevention of high frozen. Yes, again this freezing method, cryo freezing method, there are two freezing methods are there. Slow freezing method, rapid freezing method. In the slow freezing method, in this process, the tissue or plant material is slowly frozen at slow cooling rate. The major advantage is that the plant cells are partially hydrated and serve in a better manner. Compare than rapid frozen method, the slow freezing method is very good. Slowly it will be freeze. The cooling rate is slow. Whereas the rapid freezing method, the whales of plunged in liquid nitrogen. In this process, the temperature decreases from the minus 300 to minus 1000 degrees. It's not safe. Always we suggest the slow freezing method. Third one is the dry freezing method. In this method, hydrated cells and seeds are stored. The next step is storage in liquid nitrogen. It's also important for the maintenance of the cell or material, a specific temperature. In general, the temperature is kept minus 70 to minus 196 degrees. Already we discussed in previous. Prolonged storage is done at the temperature of minus 196 degrees in liquid nitrogen. A continuous supply of nitrogen is needed to prevent damage. So first of all is the selection is very important to rear and breed, which is the a specific character to produce recopies in futures. The selection process is important. Then we will go to the addition of cryoprotectants. When we add the cryoprotectants, later we will keep the material in the liquid nitrogen at the temperature of minus 70 to minus 196 degrees. How many years we can preserve, we can keep in this liquid nitrogen? It's our wish. After completion, after a process will be done, then we can take the material and we will go to the thawing process. Thawing process nothing but until the material is in state is very frozen state. Later we will keep the material, we will isolate the material and give the slow temperature with the warm water bath vigorously. Then it also causes the whales get transferred or move another bath. What is the importance of thawing? Yes, the thawing process is usually carried out by the plunging the whales. After thawing, we have to wash the material. Clearly, we will wash the material, remove the cryoprotectants, which is attaches to the material. Furthermore, the material is recultured in a fresh medium. Reculture. After thawing, we have to wash the material carefully, and then again we can reculturing. After reculturing, measurement of viability is very important. Viability is nothing but the potentiality of the ability of the material, how can it grows. Due to storage stress, there is a possibility of cell death. The presence of viability can be seen in the most cases. It is calculated by the formula. After viability, it will go to the regeneration of plants and animals. After that, the viable seeds are cultured on a non-specific growth medium. Suitable environmental conditions are maintained. So we have to keep in mind all those steps which is we had the steps in cryoprotectants. Isolation, selecting, adding of cryoprotectant, keeping the liquid nitrogen, thawing process, reculturing process, test the viability. So, what are the applications of cryoprotectants? We can see now. It is an ideal method for the long-term conservation of material. Long-term conservation methods clearly mentioned here. Disease-free plants can be conserved and propagated, and seed can be maintained for a long time. Endangered species can be maintained. Endangered species also can be maintained through the cryopreservation. Pollens can be maintained to increase the longevity. Long-lasting, long life, endangered species can reculture through the cryopreservation process. Yes, some disadvantages also there, but we will give importance to the only applications and advantages. Thank you, my dear students.